Hello, everyone. Welcome to today's webinar. I'm Pat Darkantis, contributing editor at C&EN Brand Lab, and I will be moderating today's event. This webinar is titled The Importance of Light Scattering in Biopharmaceutical Formulation Development and is being sponsored by Wyatt Technology. Wyatt Technology is the recognized leader in instrumentation for determining the absolute molar mass, size, charge, and interactions of macromolecules and nanoparticles in solution. During the presentation, we will hear from Dr. Mark Spears, Jr., a senior scientist at Fujifilm Diosynth Biotechnologies in Research Triangle Park, North Carolina. He earned a PhD from Georgia Tech in 2014, where his work focused on biocompatible polymer microstructures and resulted in several first author publications. Mark previously worked at Wyatt Technology as an application scientist. He developed expertise in light scattering techniques and biophysical characterization of polymers and proteins. He also supported business development and sales activity in the southeastern United States. In 2018, Mark joined the Formulation Development Group at Fujifilm, where he works primarily on protein formulations, including analytical characterization and innovation. I will now hand over to Dr. Spears.
Hello, everyone, and thank you for tuning in. I'd like to start with a thank you to Wyatt Technology for sponsoring today's talk, and a thank you to C&EN for hosting today's webinar. I'm honored to present today's topic, which is the importance of light scattering in biopharmaceutical formulation development. First, a little bit about us as a company. We have 3 sites in total, 2 sites in the United States and 1 in the UK. We have a little more than 1,200 people and a huge amount of experience developing and manufacturing biologics. Actually, it says 25 plus. We're getting close to 30 years now. We've worked on more than 300 molecules. We have 6 commercial manufacturing licenses and offer a full range of development and manufacturing capabilities. Now, the group that I'm part of is located in Research Triangle Park, North Carolina, and it's a fantastic place to live. So if you're looking to make a move, give it a shot. But my group focuses solely on protein formulation development. So the outline for today's talk looks like this. First, I'll give you a brief intro to what formulation development is all about. What do we mean by that term? Why should you care about it, etc. Then we'll open up our analytical toolbox here at Fujifilm and talk in particular about the light scattering tools that we are putting to use. Third, I'll briefly cover a couple slides about the typical workflows that we use in our group. And then the largest part of the talk will be the 3 different case studies where I will show you how light scattering data made a huge difference in how we understood each molecule, and in some circumstances was the only way to accurately measure a sample. Okay, so let's dive into part 1, an introduction to formulation development. So what do we mean when we use the term formulation development anyway? Well, it all starts with this basic understanding that proteins have complex primary, secondary, and tertiary structures that determine their function, and that every protein is slightly different, even within the same structural and molecular class. So our goal as formulation development scientists is to identify either a solution or lyophilization condition that will stabilize the protein throughout its entire lifetime and intended use. And that really means we're trying to preserve its molecular structure and make sure that its level of activity, which is closely related to that structure, does not change, even through stressful conditions like high temperatures, physical agitation, or even the type of container in which the sample is stored. Because we're a contract development and manufacturing organization, we encounter a very diverse set of proteins. That keeps my job really interesting, but it also presents some challenges. The breakdown of our projects is usually about 60% monoclonal antibodies, and then about 40% everything else you can possibly imagine. Typically, a molecule will come to us already in its final form, so we usually don't have any options to change the properties of the protein itself. So our job is to develop a formulation for the molecule as it already exists when it arrives at our facility. Now, our lab is non-GMP, and I'm thankful for that because we not only avoid some of the bureaucratic headaches that come with GMP work, but we also have the flexibility to tailor experimental conditions, try novel things in our lab, and also innovate in partnership with our various clients. Of course, we're still bound by project timelines as contracted with our customers, but we do have some more flexibility than our GMP counterparts. Okay, so that's the 40,000-foot view of what my group does at Fujifilm, what formulation development is. We believe strongly that formulation development is a crucial part of developing a protein therapeutic, and hopefully the case studies that I will go over will convince you of that in just a few short slides. Let's move on to part 2 about our analytical toolbox here at Fujifilm. When we study a molecule, we take a holistic approach, which means we apply as many analytical assays to the molecule as makes good scientific sense. Then we take all of those data together to paint a picture of the molecule's behavior and personality. And we have many tools at our disposal. You can see a partial list here, which represents a combination of biophysical and physiochemical methods spread across the formulations group and our analytical development group. We have set up our lab so that many samples can be prepared robotically and then transferred to the instrument for analysis, and many of our instruments are plate-based, high-throughput type of setups, so we can handle a large number of samples and screen many different formulation conditions. There's a huge amount I could say about the robotics capabilities in our lab. If you're interested, contact me later with any questions. But what I really want to do today is focus on the light scattering tools in our lab. And you can see those tools highlighted here in this orange color. We use dynamic light scattering, size exclusion chromatography combined with multi-angle light scattering detection, and refractive index detection. And by the way, I should say that we have multi-angle light scattering and RI detectors both on our HPLC and our UPLC systems. They actually require slightly different kinds of MALS and RI detectors. And we're also doing concentration gradient multi-angle light scattering, or CG MALS. I'll talk a little bit about each one of these techniques in the next few slides. First is DLS. We own the Wyatt Dynapro Plate Reader 3, which is probably my favorite instrument in our lab. It is a plate-based dynamic light scattering instrument. It allows you to use commercially available well plates and make the measurement directly in the well plate without any liquid transfer. Typically, we're using 384 well plates. We do that because the volume requirement is low and we can get a lot of sample reads out of a single plate. But we've also started playing around with 96 well plates. The reason for that is that so sample prep can be automated and take some of the laborious tasks of preparing a well plate out of our hands. DLS is important for our work because we can measure the sample's hydrodynamic radius and polydispersity as a way to track stability over time. We also use it for detection of aggregate populations. Now, most of our drug substances are very super clean, so we end up with a single monodispersed peak in DLS, but on occasion we do see aggregate peaks, or we see shifts in the average size as higher order oligomers form under stress conditions. We're routinely measuring concentration series with DLS as well to understand the strength of attractive or repulsive intermolecular forces in solution, and we're doing either isothermal measurements or temperature scans to monitor the unfolding and aggregation temperature of our samples. This last technique that I just mentioned is similar to differential scanning calorimetry, but the readout is hydrodynamic size instead of energy input. We're using our DLS plate reader very, very heavily for our formulation programs. The next technique is size exclusion chromatography with multi-angle light scattering, or SEC MALS. Most of you are probably familiar with SEC, but not as familiar with adding the light scattering detection portion of the system. The system looks identical to a normal SEC setup, except you add light scattering and refractive index detectors on the back end. You simply inject the sample just like normal, and the sample travels through the UV detector, and then instead of going to waste, it goes through light scattering and RI. And so now, instead of just the UV signal, you're actually collecting 3 signals in total. The UV, light scattering, and RI signals all together at one time. Both the UV and RI provide a concentration signal, and the light scattering detector measures scattering intensity, as you might expect. Those pieces of information together allow you to calculate absolute molecular weights. Now, the beautiful thing about this setup is that the molecular weight you measure is completely unrelated to retention time. One of the case studies later in the talk will show you why this is so incredibly important. And we're using SEC models to measure the absolute molecular weight of the main peak and any aggregates or fragments that may form in solution. And we can track these parameters over time as the sample is subjected to stress or is exchanged into different formulations. There's so much more you can do with an SEC mols instrument setup. One example that I'll show you later in the talk is how we measure a glycosylated molecule, and that will play a vital role in one of our case studies today. Last but not least is concentration gradient multi-angle light scattering. This experiment uses the same mols detector that was on the previous slide for SEC mols, but there are some differences in the way the experiment is conducted. We're either using the Wyatt Calypso instrument to automatically prepare a concentration series, or we manually fill a cuvette, measure, and then clean it and move on to the next concentration. The automated way with the Calypso is a little bit easier, and the data tend to be a little bit cleaner, but it takes a lot more sample. The cuvette way is more labor intensive, but uses only a tiny, tiny amount of sample, so often we end up using the cuvette. The experiment works like this. We prepare a concentration series from our target concentration all the way down to about 1 mg per mil or less, and the light scattering intensity is measured at each one of the concentrations. Then the results are fit to a reversible association model, and we're able to get the strength of self-association and potentially some molecular stoichiometry across the entire concentration range. We've found this very informative, not only to measure the strength of attractive or repulsive interactions in the solution, but also to measure the percent monomer across the concentration range. What's neat is we can tell exactly where this percentage dips below 95% monomer and therefore begins to fail the regulatory requirement for purity. Okay, so those are the light scattering techniques that we're currently employing in our lab. So let's move on to part 3 about our typical workflows. We offer pretty much the whole spectrum of formulation development, which might include some or all of what you see here on this slide. Traditionally, each program we bring on board gets a custom designed formulation development study. And there are basically 4 primary parts to this design. There's pre-formulation development, which is the launching point for most programs with biophysical characterization, molecular modeling, and some understanding of the upstream and downstream and filtration processing effects. In this phase, we're trying to understand what we term the, quote, personality of the molecule. Then we move on to the second part, which is the actual formulation development, and there's a huge amount of work that fits within this part. Everything from studying the effects of pH, buffers, ionic strength, and excipients, all the way through container closure studies, route of administration investigations, and so forth. Then we move on to real-time stability and accelerated stability, which includes monitoring the stability of the sample over a long period of time. And what we're really trying to do there is show regulatory agencies that we can substantiate our claimed expiry date and the storage conditions. Last, we also provide analytical support through preclinical, clinical, and commercial phases. That might include things like troubleshooting, verification of analytical assays, etc. Now, this slide represents the traditional way of designing a formulation development program. It could be all of these or some number of these things that you see on this slide. And if you have the resources and time, lots of resources and lots of time, it's a fantastic approach. I liked this quote by Nick Saban. He said, "A good process produces good results." For any of our international viewers or folks who are not sports fans, Nick Saban is a very successful football coach who is known for his process of developing players into highly skilled and a highly disciplined football team. So the combination of activities in points 1 and 2 on this slide usually takes about 6 months or so. And if you add in real-time and accelerated stability studies, well, the formulation development phase could last up to 2 years. But sometimes resources and time are more limited. So we also offer a more streamlined formulation development program, such as what you see here. We can move through this process in 4 distinct stages. Stage 1 is the pre-formulation personality assessment, and that's similar to point 1 on the previous slide, with a few extra little stress tests thrown in. Then we narrow down to stage 2, which is a combined pH, buffer, and excipient screen. We narrow down even further to stage 3, which is a predictive solubility study on only a few select conditions. And last, we narrow down to stage 4, which is accelerated and real-time stability, if requested, on only the top 1 or 2 formulation candidates. This is a very streamlined version of formulation development. And what's really exciting is we're able to get to a final formulation decision in 16 weeks. Our real-time stability results will not be complete at that particular point in time, but we will have enough data to make an informed decision after 16 weeks. And light scattering data is used in every single stage of not only the custom process on the previous slide, but this more streamlined process as well. Now, my manager, Catherine Bowers, presented a webinar for Wyatt back in 2015, and I blatantly stole a slide from her talk, and that's what I'm showing you here. I found it really interesting because she was pointing out at that time not only all of the many hats which formulation development scientists wear and how we touch on so many areas of the business, but she also included this note in yellow on the right side where she dreamed about and predicted a day at Fujifilm when light scattering techniques would be applied to virtually every molecule being developed in our facility. And I brought the slide back because I think we're there. Light scattering has played a vital role in the development of every molecule that I have touched since I've been a part of Fujifilm. And we've built light scattering analysis directly into our standard workflows. So I really believe we're seeing the fulfillment of Catherine's predictions from all the way back in 2015. And now I'd like to show you some real examples of how that's true. And I'll do that in part 4 of the talk, and this will be our case studies. We'll spend the majority of our time here in this section of the talk. This part will be split into 3 different distinct case studies, 3 different projects that have come through our labs. So let me start with some background for case study number 1. Now, it's hard to believe, but this molecule was studied by another contract organization for 10 years, and very little progress was made during that time. In fairness, though, it is a very tricky molecule. It's known to produce higher order aggregates during the processing steps. It's also highly glycosylated. To give you an idea of molecular weights, the protein by itself, without any glycosylation, is 89 kilodaltons. And then when you add in the glycosylation, it can be anywhere from around 100 to 130 kilodaltons, depending on the maturity level of the glycans. When this molecule came to us, the client was unsure of a lot of things. They were unsure of any correlation between the molecule's activity and its aggregation state. They had no way of knowing what its activity was like relative to the method of expression and purification. And they didn't even necessarily know the active form of the molecule. At that time, the client thought that the active form was a pentamer, but it was really hard to tell because of ambiguity in the various activity analyses that were being performed. So stick that in the back of your mind. The client thought it was a pentamer. The active form was a pentamer when it came to us. So the program started up. We received 2 lots of material, and I'll just call them X and Y for simplicity. Lot X was less pure, but it was active. And lot Y was nice and pure, but it was inactive. Okay, so I'll refer to X and Y on the next couple slides. So just keep that in the back of your mind as well. X is active, Y is inactive. We used SEC mols to identify the main peak molecular weight. Remember that SEC mols gives us the absolute molecular weight regardless of elution time. Here you see 2 different chromatograms for X and Y. The light scattering signal is the solid blue and solid red lines, and then the measured molar mass is the dotted lines overlaid on each figure. So for lot X, you can clearly see that the molar mass is close to 300 kilodaltons. And for lot Y, you can see that the molar mass is close to 500 kilodaltons. Now, if you had to guess based on these initial results, you would probably say, okay, lot X is likely a trimer, and lot Y is likely a pentamer, and you would be right. But remember, this challenged the client's belief. The client believed that the active form was a pentamer, but lot Y, which is a pentamer, was inactive. And we were able to confirm this with SEC mols. Lot X is the trimeric form, but it's active. So this was an unexpected result, and it took some convincing both internally and externally to get folks to buy into these data. Once we had established that the active form was trimeric, we moved on to look at the glycan content. We performed glycan analysis using mass spec on both the initial lots and multiple lots of material that we produced here at our own facility. And we determined that the glycan mass, the total glycan mass, was actually unrelated to activity. So for example, we could have 2 different lots that had different levels of glycosylation, but they had the same amount of activity. So the total glycan mass was unrelated to activity. Now, mass spec glycan analysis requires removal of the glycans from the molecule. But what's great about SEC mols is we can actually measure glycan content on the intact molecule during a normal chromatography run. So when a molecule is glycosylated, it does complicate the analysis a little bit. So we have to use some more advanced data processing, which we call a protein conjugate analysis. And I'm going to push pause on the case study just for one slide so I can show you what protein conjugate analysis is all about, and then we'll go right back to case study number 1. So I borrowed a beautiful glycosylated protein example from Wyatt for this slide, so thank you, Wyatt. To do this analysis, you must perform SEC mols, and you must collect all 3 of the signals that I described before. So UV, light scattering, and refractive index. With the UV and light scattering signals, you can calculate the molecular weight of the protein portion only. With the refractive index and light scattering signals, you can calculate the molecular weight of the total molecule. And then you simply subtract the protein from the total, and you get the glycan molecular weight. Now, I'm simplifying a little bit. The math is actually a little bit more complicated that goes on in the background. But once you know the molecular weight of each part of the molecule, it's actually just simple math to calculate the mass fractions as well. Now, the graph looks something like what you see here. For each peak, you see 3 different lines. The top, more solid line is the total molecular weight. The middle line is the protein molecular weight. And then the bottom line is the glycan molecular weight. Now, this is a very powerful analysis. For every single peak in the chromatogram, you're measuring the composition of the molecule and even potentially changes in the composition across the peak. By the way, I should mention this protein conjugate analysis works for antibody-drug conjugates, also works for pegylated molecules, and just in case there are any polymer scientists in the audience today, it also works for some copolymers in some cases. Okay, so that's what we're doing. That's the kind of analysis that we're applying to this molecule. So back to the case study. We used the protein conjugate analysis not only on those initial X and Y lots, but also on additional lots that we made at Fujifilm. For simplicity, I'm only showing results for the 5 most recent lots. The graph on the left is the result from one of these lots. So this is just a representative example. And you can see it looks very similar to the graph from the previous slide. The total molecular weight is on the top. The protein molecular weight's in the middle, and then the glycan is on the bottom. And so if you look at the table for 1 second, I'd like to work across the table. So the average total molecular weight in the first column is 325 kilodaltons. And the molecular weight across this peak is actually very monodisperse. You can see our polydispersity number there is 1.000. So this is a very monodispersed pure sample. The protein fraction percentage is 79.2%, and that is 257 kilodaltons. So of the 325, 257 is protein content. And then the glycan content is about 68 kilodaltons. Notice that these results are consistent from lot to lot. So if you work your way down the table, you're seeing very good consistency from lot to lot. And that's a fairly recent significant achievement for this particular tricky molecule. And also remember that this molecule is a trimer. So if you were to take that 257 kilodalton number for the protein molecular weight and divide that by 3, then you get 86 kilodaltons, which very closely matches the theoretical value of this protein monomer, which, if you remember from the first slide, was 89 kilodaltons. So to summarize case study 1, we used SEC mols to confirm that the active form of this molecule was actually a trimer. I didn't give you all the information, but early in the program, the glycan content was actually much smaller, about half of the expected mass per site. And now we've been able to produce drug substance with mature glycans, and we're also able to consistently produce drug substance with essentially 100% trimeric form of the molecule, and we can track it through production, with light scattering data, SEC mols, and the protein conjugate analysis. Okay, let's move on to case study number 2. This product is a mixture of 3 proteins, and each has nearly an identical theoretical molecular weight. We'll call these 3 proteins ABC for simplicity. Each one of these proteins is expressed individually, and then after expression, they're combined in a 1:1:1 ratio, and the total protein concentration is kept constant. Now, obviously, this product is going to be an analytical challenge because of its complexity, and because these things are going to be in a mixture. One of the main questions we had going into the project was, is it even possible to assess the sample purity in the mixture? Could we even distinguish impurities from the sample itself with such a complex background and a complex type of sample? So we were tasked with pre-formulation and formulation development studies on the molecules individually, and then formulation development on the molecules in combination as well. Now, obviously, we collected lots of other types of data, but I'm going to focus in on the SEC mols portion. First, we injected each molecule individually on a UPLC, and these are the elution profiles for A, B, and C, plotted versus time on a relative scale. Now, if you're tracking with me, you'll see an immediate issue with these data. These proteins have nearly the same molecular weight, but they have very different retention times. And if you were to assign a molecular weight based on a standard calibration curve, you would get numbers that just don't make any sense. Molecule A would be 166 kilodaltons, molecule B would be 36 kilodaltons, and molecule C would be 17 kilodaltons. Now, obviously, this is a problem. So based on some other data that I'm not showing you here on this slide, we were able to determine that the difference in elution time is simply due to interaction with the column. But never fear, because we have light scattering, which will give us the correct molecular weight regardless of elution time. The molar mass has been overlaid on the light scattering chromatogram, and you can see that the main peak for each one of these is right at about 150 kilodaltons, and each sample has a small amount of high molecular weight species at the 4.2, 4.5, and 5.2 minute marks. Most of those look like dimer. In the case of sample C, there is also a small peak at 5.4 minutes, somewhere between the monomer and dimer. We were able to determine that for each of these samples, the impurities are less than 5% of the mass fraction of the sample. We weren't really sure if the mixture would have the same elution profile as the individual molecules, but it turns out that the profile looks very similar. The light scattering trace for the mixture is in red, and it's overlaid on top of the individual light scattering traces. You can see the main peaks actually overlay very nicely. Now, the light scattering signal is a little bit smaller in the combined case, but remember that the total protein concentration was held constant. So the content of each individual molecule in this combined sample is actually 1/3 of the concentration of its original injection. Here I have removed the results for the individual molecules and overlaid the molar mass results for only the mixture. The main peak molecular weights are still 150 kilodaltons. We do see some impurities still. There is a dimer peak still at about 4.2 minutes. We really don't see those impurities between the A and B peaks like before. We're seeing something around what looks like 150 kilodaltons or so eluting in that space between A and B. We don't really know if this is maybe an average of small species from A and large species from B that average out to about 150. We don't actually know for sure what those impurities are, or if they're even still present in the mixture. Since the proteins have been diluted by 1/3, it's also possible that some of the oligomers and other impurities have actually been diluted out. Now, finally, if we overlay the molar mass results from the individual runs and the mixture run, you can see that the main peaks overlay perfectly, while the impurities show some differences. So we're still seeing the dimer at 4.2 minutes, as I pointed out before. It seems that some of the high molecular weight peaks get lost once the molecules are mixed. For example, the blue peak at 5.4 minutes is simply gone in the mixture run. That might be because it's co-eluting with the front of the main peak for B. So perhaps the signal is almost entirely due at that point to the main peak species. And that could be the case or the same situation for the green peak at 4.5 minutes. It could be eluting with the front of the peak, the main peak for molecule A. But we were able to see some impurities still, even in the mixture. So to summarize, this was a very challenging sample. Although we were able to get accurate molecular weights for similar species eluting at different times, both as individual molecules and in the mixture, there's no way we could have determined this without light scattering data. There's just absolutely no way we could have relied on elution time in this case. We did seem to lose some resolution of impurities, but as far as we can tell, the mass fraction of the impurities seemed to be low, even in the case of the mixture. Okay, last but not least is case study number 3. This molecule is quite different from the other molecules I've presented thus far. This molecule has a very unusual structure with multiple arms branching out from a central hub, kind of like spokes of a wheel, except in 3 dimensions. The number of arms can vary. It's usually no less than 4, but it can be many more than that. The native form of this molecule is thought to be a dodecamer with a total molar mass of 512 kilodaltons. A dodecamer is actually 12 subunits, if you're not familiar with that term. So I'll let you do the math and get the molecular weight of each protein monomer from that knowledge. So the size and molecular weight of this molecule, as you might imagine, did present some challenges with traditional protein analysis methods. Now, my colleagues ran some size exclusion chromatography on this material, and they used a really special column set to try and resolve these huge species from possible low molecular weight species that might exist in the solution. And of course, what you're seeing here is the UV trace. With this method, we saw the first peak eluting at about 10.6 minutes and the last peak eluting at 20.6 minutes with the void peak at around 25 minutes. Now, some assumptions were made about assignment of the peaks, as you can see here from these labels. The first peak was thought to be an aggregate of some kind. The dodecamer was assigned to the second peak. And several low molecular weight peaks were identified as well, but not assigned to any particular species. After this chromatogram was collected, my colleagues made several changes to the method to try and improve resolution and mobile phase and so forth. So then I ran SEC with their new and improved method. And this is what I got. So you can see immediately from this graph there's some significant differences in the method and also the peak shape and things like that. The solid red line is the UV signal, and the dotted red line is the molar mass. So the first big peak is not resolved, and now it just looks like a front shoulder around 23 minutes. The main peak, which my colleagues identified as dodecamer, is right at 25 minutes. And then there are a couple low molecular weight peaks at 30 and 32 minutes. Because we are using SEC mols, we can accurately measure the absolute molecular weight of these peaks and not have to guess about peak assignments, which is great. So interestingly, the lowest molecular weight we measured is about 760 kilodaltons, which is well above the presumed dodecamer mass of 512 kilodaltons. And even the so-called low molecular weight peaks are actually not low molecular weight. The molecular weight stays fairly even and constant across these peaks, even though they're eluting later in the chromatogram. This analysis was done at 20 degrees Celsius. Now that's not very meaningful for this particular slide that we're looking at, but it will come into play in a few slides. So just stick that in the back of your brain for a second, and we'll come back to it. Based on this SEC mols analysis, we concluded that there's likely no dodecamer in the sample. It's just not there, or it's at least not resolved from the other species. It's not something we can see by this particular technique. I created a cumulative weight fraction graph of these same data. Now this plot has molar mass on the x-axis and cumulative weight fraction on the y-axis. If you're unfamiliar, the way to read this is at a particular point on the curve, the fraction of the sample is at or below the corresponding molecular weight. So for example, if you look at the gray dot, only 20 to 25% of the sample is less than or equal to 1 million daltons. Or if we flip that around, the great majority of the sample, about 75 to 80%, has a molar mass above 1 million daltons. So the molar mass of the sample is far greater than initially was hoped for or assigned based on SEC results. Now, obviously, this sample is not as well behaved as we first thought, and it needed some further investigation. So we decided to analyze the sample with differential scanning calorimetry using a temperature scan from 10 to 110 degrees Celsius. And we observed 3 transitions over a temperature range from 21 to 45 degrees. We were actually shocked to see that this molecule has an onset temperature of 21.6 degrees Celsius. So in other words, the molecule is unstable even at room temperature. So even just letting a sample thaw on the lab bench could be altering the sample's integrity. Based on this revelation, we needed to reevaluate our SEC and SEC mols results, and we needed to collect DLS data at different temperatures to see what changes were happening to the molecular size. We did a temperature scan with the DLS from 10 to 60 degrees Celsius. And the first thing to notice is that the starting size is very large. The starting radius is 54 nanometers. So think about that. The total diameter of this particle is over 100 nanometers in size. At this point, we've really left the protein dimension, and we're in the nanoparticle world. And so we know from our SEC and SEC mols results that this size is an average of a range of species in solution, right? High and low molecular weights. And we'll simply treat it here as an average size for simplicity. So as you increase the temperature and continue taking DLS measurements along the way, the size remains constant up to about 25 degrees. And then the size begins decreasing all the way down to 30 nanometers in radius at a temperature of 42 degrees. And then above 44 degrees or so, the sample aggregates uncontrollably, and the aggregation is not reversible. We did test that, even though I'm not showing those data here. This is really interesting behavior where the particle seems to collapse as the temperature goes up. But at DLS, we're seeing size changes around the same temperature range as we saw with DSC, which is great. The two techniques seem to talk to each other. And clearly, the temperature at which the analysis is conducted is an important parameter for this sample. So we went back and reran our SEC mols test at 3 different temperatures, 20, 25, and 30 degrees Celsius. And again, what you're seeing here is the UV signal as the solid lines and the molar mass as the more dotted lines on top. There are definitely differences in the elution profiles at the different temperatures. The main peak shifts to later time as the temperature goes up, which also causes the front shoulder around 23 degrees to be more pronounced at higher temperatures. We also see higher absorbance of the low molecular weight peaks at higher temperature, as you can see here from the green trace. The molar mass distribution is very interesting to compare between these 3 runs. We see generally the same curve shape with very high molecular weight species eluting first, then some lower molar mass species eluting later in the run. And you get a small bump in molar mass that maps onto the UV signal. There are some small differences in the high and intermediate molecular weight species, but the primary differences are in the lower molar mass region from about 28 to 34 minutes. As the temperature goes up, the molar mass goes down. And we've already seen that the red curve, where the majority of the sample is above 1 million Daltons, and the minimum was about 760. But take a look at the green curve. At 30 degrees, the average molecular weight in that lower region from 28 to 34 minutes is right at about 500 kilodaltons, which fits very well with our dodecamer prediction. Now the blue curve lies somewhere between those 2 signals, both in intensity and molar mass. You can easily see the different molar mass distributions in the cumulative weight fraction graph as well. The percentage of the sample with low molar mass goes up as the temperature goes up. This plot confirms what we thought from the previous slide and also adds to our understanding by giving us the actual percentage of the sample at each molar mass across the distribution. So to recap case study number 3, this is an ongoing investigation. There's quite a bit more we feel we need to learn about this sample. We don't yet claim to fully understand the molecule's behavior, but we've certainly gotten a good start with our light scattering analysis. SEC mols confirmed that the sample is not in the dodecamer form which we initially thought. And dynamic light scattering and DSC data taken together do provide some insight into the behavior of the sample at different temperatures and revealed the importance of paying attention to the analysis temperature as we go along. And finally, SEC mols at the different temperatures revealed some pretty significant changes in molar mass distributions. And if we didn't have this equipment or did not run SEC mols, I think we would have blindly and incorrectly assigned SEC peaks to different species. To summarize today's talk, our formulation development group at Fujifilm relies heavily on our light scattering equipment as a crucial part of our analytical toolbox. And it touches on virtually every part or stage of our workflows and has played an important role in many of our projects, such as these 3 case studies that we've covered today. And we think that moving forward, our group will continue to lean heavily on light scattering data in combination with other analytical and biophysical tools for fully characterizing each new molecule and comparing formulations one against another. I'd like to acknowledge some of the folks from our group who have contributed to these 3 case studies. Wyatt Technology is the manufacturer of all the light scattering equipment that I've talked about, and they have provided us some very good technical support as we encounter all kinds of crazy new molecules and interesting light scattering data from those molecules. And finally, I'll say a thank you to you, the listener, for tuning in and for all the time you've given and dedicated to learn about our work. And I would be delighted to answer questions about today's presentation.
Hello, this first question is a 2-part question. Number 1, how can you separate the sample from the void peak and from the solvent RI signal? And 2, how can you best separate out a complex mixture with a broad polydispersity on sec mols that are not separating out enough using 2 columns?
Yeah, so we are dependent on good chromatography. So your multi-angle light scattering measurement is only going to be as good as the purity of the peaks that are coming off of your chromatography system. So we are dependent on a good column selection and a good mobile phase selection so that the peaks elute well before the solvent RI signal. So that separation just happens based on the column selection and mobile phase selection. And in terms of separating out broad polydispersed samples, so again, we are dependent on the column and good chromatography, but multi-angle light scattering will also pick up on a very polydispersed sample. So it will give you the molecular weight range from high to low. So we are dependent on good chromatography, but we can also analyze a very polydispersed sample that's coming off.
Mm-hmm.
I should also mention that in some cases, particularly if this question is relating to polymers, there are other separation techniques that can be combined with multi-angle light scattering. So there's a technique called field flow fractionation, and if for some reason your sample is not amenable to using a column or has some kind of interaction with the stationary phase, you can also use field flow fractionation as a separation technique and then run the sample through light scattering detection and make essentially the same molecular weight measurement on the back end.
Great. The next question is regarding case study number 1. Is glycosylation known to promote aggregation or instability for these types of molecules, or does it have some other role?
Yeah, good question. So sometimes glycosylation does promote instability, and it really just depends on the particular molecule. But often we also see that glycosylation helps to regulate the interaction with the receptor. So it plays some kind of role in the biology that's happening for that particular molecule.
Mm-hmm. Great. And for conjugate analysis, what DNDC value did you use for the glycan?
So we used 0.13. It really depends on the particular, if you have some kind of knowledge about the glycan, But typically, 0.13 is a good value to use for glycans in the conjugate analysis. And then, of course, the protein, the NDC is 0.185. And when you plug both of those values in the correct spots, the Astra software will do the calculation for you based on the weight percentage of both parts of the molecule, and then automatically input the correct DNDC value to use in the molecular weight calculation.
Mm-hmm. And the next question is, what is the largest size detectable by moles?
Yeah, so multi-angle light scattering, we are dependent on, or there's an influence on the number of angles that we're using to measure. So the number of angles determines both the upper size and also upper molecular weight that we can accurately measure. In our case, we're using an 18-angle instrument, and that allows us to measure sizes up to 1 micron and molecular weights up to about 1 gigadalton. So extremely large molecules, but essentially anything up to a micron in size we can measure accurately, both molecular weight and the size as well.
Great. And the next question is, both DLS and CG moles could measure attractive and repulsive interaction. Which one has better resolution and accuracy, and how do you choose that?
Yeah, that's a great question. So fundamentally, slightly different things. So if you're measuring attractive and repulsive interactions by DLS, it's a number or a parameter called KD, the diffusion interaction parameter. And if you're measuring attractive and repulsive forces by CG moles, then you're measuring a number called A2. And so, or some people refer to that as B22. They're very, very similar numbers. There are very slight differences between the two. Of course, the plate reader that I mentioned, the DLS plate reader, actually can do both of those at the same time. So it measures dynamic light scattering and static light scattering. Generally speaking, if you're looking for the best resolution and best accuracy, you would do CG moles, or concentration gradient moles, and you would put that into a dedicated multi-angle light scattering detector. That's where you're going to get the best resolution and best accuracy over a really wide concentration range. The advantage of DLS is its speed. So you can measure KD and also measure A2 very, very quickly. You sacrifice a little bit of resolution, but you gain efficiency and speed.
Mm-hmm. And the next question we have is, for the conjugation analysis for moles, is this a software feature or is an additional analytical run needed on SEC moles?
So this is a built-in software feature. It is a protocol that's already built into the software immediately upon installation. And what's really beautiful about this is you can apply that analysis to essentially any chromatography run. So you don't have to do any additional experimentation. You don't have to do any additional runs or anything like that. You just simply apply the analysis to the data you've already collected. So it's really quite easy to do from that standpoint.
Mm-hmm. Great. And the next question is, can SEC moles analyze the molecule of case study number 3 to determine the number of arms branching out from the central hub?
Sure. Yeah. So case study number 3, just as a quick reminder, was the central hub when the spokes kind of branching out, really large molecule. And so what's interesting is that molecule in our hands is acting actually more like a polymer than it is a protein. And those who have done polymer work and GPC work in their life will recognize immediately that we can do some branching analysis. So there is actually branching analysis already built into the software as well. That would speak to determining the number of arms branching out from the hub. And the other thing we can do here is we know the molecular weight of each individual arm. So if we're able to calculate the total molecular weight, we can simply divide that by the number or the molecular weight of each arm and figure out the stoichiometry and the number of arms that are branching out.
Mm-hmm. Great. And in case study number 3, the DLS data show a decrease in radius with temperature of the nominally quite large molecules. Can you ascertain from the DLS measurement if the molecule is dissociating or simply changing conformation?
Sure. So as we collect dynamic light scattering, we are not only measuring a size, but the software is also collecting the count rate. So the count rate is literally just the number of photons that are scattering off of your sample. And for light scattering, something that is twice the mass will scatter twice the amount of light. And so you would expect the count rate to be double. And also, if we were to either start to form aggregates, or if the molecule was starting to break up, we would see the count rate go up or go down, respectively. In the case of this case study, the count rate actually stayed constant throughout the entire temperature scan until we got to that point where it irreversibly aggregated and the size really shot off. But in the regions where it was simply we're seeing a decrease in size, the count rate stayed constant. So we believe that we're seeing a conformational change, not a dissociation event, or the molecule staying intact. It's simply changing conformation.
Excellent. And my next question, the lot-to-lot variation of molar mass and protein fraction that you presented in case study number 1 was about 1 to 2%. How much of this is from experimental repeatability, and how much of this is actual product variation?
So there are a lot of factors that go into this measurement. And there's a lot of variation in, because this molecule is highly glycosylated, there's a lot of variation in the glycan content. There is lot-to-lot variation that just naturally occurs, and there is some experimental variation that's built in. We're also doing protein conjugate analysis, which means we're trying to tease out sort of smaller parts of the molecule, which also introduces another variable. Generally speaking, and this is just kind of a rule of thumb, the repeatability in a SEC mols experiment is the variation is less than 1%. So it's very repeatable. Here we're seeing 1 to 2% or so between lot to lot, and so I think that we are definitely seeing a slight amount of product variation. But again, there are lots of factors that are going into the measurement, and it's kind of difficult to tease out exactly what what is due to the lot-to-lot variation and processing conditions, and what's simply due to the measurement.
Great. Well, that's all the time we have. So thank you again, Dr. Spears, for your fascinating presentation. And thank you, participants, for being a great audience. Goodbye.